Hi friends, welcome to my channel Absolute Biopharma. I am your host Pallavi. So today's topic for this video is fermentation. This is my first video for the bioreactor series. So if you are new to this channel, then do watch my previous videos so that you will get an idea about what this channel is. So without any further ado, let's begin the video. So let us see the contents of this video. Stages of bioprocessing, upstream bioprocessing, what is fermentation, why fermentation with living materials, process of fermentation, steps involved in fermentation, and the limitations of fermentation. So let's begin the video. To understand the fermentation, first let us see the stages of bioprocessing. There are two stages. First is upstream processing, second is downstream processing. What is upstream processing? If you can see in the figure, there are various steps in upstream processing. It starts from clone selection, media optimization, cell banking, vial thawing, fermentation and harvest. In this video, I am only going to cover the topic of fermentation. If you want to know about cell banking, you can click on the I button and you can watch the video on cell banking. So the person who gave the term fermentation is Louis Pasteur. So some of the products that we can get from fermentation are enzymes, amino acids, organic acids, vitamins, antibiotics, solvents and fuels. So now let us see what is fermentation. If you see in the figure, the simple definition is utilizing the living cells to get the desired product is fermentation. So in the terms of bioprocessing, any process that uses living cells or their components to obtain the desired product is fermentation. Why fermentation is done with living materials? Complex molecules such as antibiotics, enzymes, vitamins are impossible to produce chemically. Optically active compounds like amino acids and organic acids are difficult to prepare chemically. Though some of the products that can be economically derived by chemical process, but for food purpose they are better if produced by fermentation, such as beverages, ethanol, vinegar, etc. Fermentation usually uses renewable feedstocks instead of petrochemicals. So let us see the process of fermentation. It includes various stages, in which first is media formulation, second sterilization, then inoculum generation, providing optimum growth conditions, generation of product, extraction of product and purification of product. Let us see each topic individually. First is media formulation. In media formulation, various types of medias and feeds are provided by the vendors and the R&D team optimizes whichever is the best media for the desired clone, and that media is used further for processing in production. Second is sterilization. Before starting fermentation, the vessel in which the process need to be carried out needs to be sterilized. In sterilization, there are two categories. First is CIP, second is SIP. CIP stands for cleaning in place and SIP stands for sterilization in place. These two aspects of cleaning are covered in my upcoming videos of bioreactor series. Third is inoculum generation. Inoculum generation is also referred as seed generation. What is a seed? Seed is a small amount of cells that are inoculated into the media through which the further division of the cells carry out. So in the figure you can see there is a 125 ml flask, there is 250 ml flask, there is 1 liter flask and 2 liter flask. From these subsequent flasks, the inoculum is increased in volume in wave bioreactor and then finally inoculated into the reactor. The reactor volume can range from 10 liter, 5 liter to 50 liter and 100 liter. It can also go up to 1 kl, 2 kl, 5 kl, 20 kl. Next is growth conditions. Once the media and the inoculum is charged into the reactor, the cells are allowed to grow in certain conditions. So those conditions can be pH, DO, air, agitation, temperature. These are the major conditions that are given. Other than that, there are many conditions that are given to the fermenter or the bioreactor for the cells to divide in best possible way. Then comes the product generation. A certain batch with a certain organism is allowed to grow for a specific period of time that can range from 12 days to 15 days according to the process to process. So that period of time from 12 days to 15 days, the cells are allowed to produce the product. This is called product generation. In product extraction, the product generated in the cell suspension is separated. The separation is done with the help of a process called depth filtration. In this process, depth filters are used. In this, the cell supernatant and the cell mass is separate. Mammalian cells will produce extracellular protein, so the product will be in the supernatant. If the fermentation is carried out with the microbial cells, the product will be intracellular and that will be produced in the cells. Next is product purification. Once the cell supernatant is separated from the cell mass, the cell supernatant undergo further purification because the cell supernatant contains multiple proteins, multiple metabolites. So to get our protein of interest or to get our product of interest, we need to purify the cell supernatant. So the figure shows both for intracellular products that are generated from microbial cells and extracellular products that are generated from mammalian cells. For extracellular products, first step is removal of insolubles that include centrifugation, flocculation, filtration, sedimentation. After removal of insolubles, isolation is carried out, which include adsorption, evaporation, extraction, precipitation, membrane filtration. Once the isolation of the product is done, it is purified through chromatography, which includes various types of chromatography, precipitation method or membrane filtration. After purification, finishing step is done. After purification, finishing is done through crystallization, lyophilization, drying or sterilization. This finishing product is not carried out for generally microbial or mammalian cells, as the product will always be in liquid form, so they are filled into the vial and get ready for final marketing. This product purification is a part of downstream processing. Let us see some of the limitations of fermentation. Products are made in complex solution in low concentration as compared to chemically derived compounds. The products generated from fermentation process are very difficult to purify. The products are expensive. The process is slow. As the process is much slower than the chemical process, there is an increase in the cost of the process. Next is process optimization, and R&D costs also make the fermentation process expensive. The waste products that have been generated from the fermentation have high level of BOD, that is biological oxygen demand, and it requires extensive sewage treatment. So that's all for this video. I hope you got an idea about what is fermentation. You can share the video with the relevant audience so that they can also get an idea about fermentation. If you want to watch this video in Hindi version, you can click on the I button and you can watch the video in Hindi. You can also suggest me the topics that you want related to the bioprocess industry or biopharmaceutical industry in the comments, so that I will get an idea what generally my audience want to see. So I will see you in my next video that will be on SCADA. Till then, take care and have a great day.